Welcome to Biotechnology number 2, where we'll now delve into the processes of PCR and gel electrophoresis, as well as recombinant DNA technologies. Biotechnology 2, so this is Pearson section 8.2. So in this lesson, we're going to describe the process of making recombinant DNA. So this is a higher level dot point than the previous lesson because it wants you to actually describe the process. So we're going to talk about restriction enzymes, plasmid vectors, and DNA ligase to do these processes, and then how bacterial transformation works. You need to explain the purpose of polymerase chain reaction, PCR, and gel electrophoresis. So again, there's nothing about explaining the processes, it just wants you to explain the purpose. And again, that same dot point, appraise data from an outcome of a current genetic biotechnology technique to determine its success rate. All right, so in terms of genetically modified and transgenic organisms, we know that humans have used selective breeding for many, many years to produce animals and plants that have ideal characteristics. So they would cross-pollinate different plants or they would crossbreed different animals to try and get ideal traits. But that relied upon a lot of trial and error and only sort of playing with the genes that were there in the gene pool. So nowadays we've got the knowledge and skills to use biotechnology to actually interfere with the normal process of breeding to be able to transfer genes from one species to another and produce new DNA combinations. So again, there's a lot of benefits to this in all sorts of fields, but there's also many ethical questions. So a GMO is an organism that's had its genome altered. So it could be by a directed mutation, it could be by new gene editing stuff like we've been talking about with CRISPR. So a GMO that has had genes from another species inserted into it is called a transgenic organism. Okay, so that's the biggest difference there, is a transgenic animal has had a gene inserted from a different species into it. So human insulin is a good example of a GMO application. So what they've done is they have previously, people who required insulin, they would obtain insulin from pigs or cows and keep that to give to the human patients. It's hard to obtain. It's not as compatible with humans as what the normal human one is. And it would, run out over time. So genetic modification can be used where the gene for the production of insulin in the human body can be isolated, placed into a bacterial plasmid, and then expressed via transcription and translation to produce insulin the way it would be produced in the human body. And so that's what you see in the image on the right there, Humulin. And it's been done in that type of way where it's very compatible with the human. It's done in a lab, so it's very efficient and it's not going to run out. So transgenic organisms, again, have a gene from another organism inserted into its genome. They've been used for many years for research, looking at diseases and potential treatments. So they will look at altering the genome of organisms. So they might insert a human gene for a particular disease into a rat so that it displays the same sort of symptoms as the human condition, and then look at how it responds to drug treatments, to look at medications and things like that. They can use knockout technology, which is where the animal model has had a gene removed to look at the role of that particular gene. They do use transgenic animals in agriculture as well. So the image on the left is a mouse carrying the gene for green fluorescent protein, GFP, which is not normally found in animals. So that's a transgenic animal. And on the right there, you've got the transgenic salmon, which would have genes inserted for growth and that sort of thing to get a lot bigger fish and feed more people. We're going to go through what this technology is all about, but this is just an overview of what genetic modification is looking like with transgenic plants at the moment using bacterium. So up the top in the blue, they're isolating the gene of interest. So this might be a gene for a particular trait that they want the plant to show. And then they get the plasmid DNA from a bacteria, which is this pink thing, and they insert the gene into that plasmid. The plasmid is taken up by the bacteria, so that's the agrobacterium, and then they put that into the plant cell and they call it a transformed plant cell. And so that plant cell now contains the gene that they were wanting it to be. It might be a gene that makes it insect resistant or produce more fruit or something like that. And then they grow that those cells in the lab on like an agar plate and produce like artificial little genetically modified plants. And then they would put them out in the field. All right, so we're going to go through the steps in making recombinant DNA. Recombinant DNA is talking about that. It is making a new combination of DNA, which is what happens in our GMOs. And the first part of that is the restriction enzyme, the cutting of the gene of interest that we're wanting to insert. So we know that DNA molecules are way too long to work with in their entirety. And in the cases of GMOs, we only want to isolate one particular gene. So a restriction enzyme is an enzyme that's able to cut DNA into smaller fragments and allow us to isolate single genes. So they're called an endonuclease. So they're going to be, it's an enzyme by the A's, and it's going to make cuts to nucleotides. The cuts take place at specific recognition sequences called restriction sites, which is different depending on the restriction enzyme that's chosen. So there's a bunch of different restriction enzymes available. They all cut DNA, but at different restriction sites. They occur naturally in bacteria and target foreign DNA to cut it and remove it. And that's how they've obtained these enzymes for use in biotechnologies. A commonly used restriction enzyme comes from E. coli and is called EcoRI or EcoR1. I'm not really sure how it's pronounced. And a model for that enzyme is shown on the left there. So you can see the DNA double helix with the pink and blue, and the enzyme is that big blob that's made a cut in that DNA. So each restriction enzyme targets a specific sequence, as I said, and it's usually 4 to 6 base pairs in length. So that allows them to select a restriction enzyme that's going to be most appropriate to isolate the gene that they want to isolate. I mentioned before that that's called a recognition site. And each time the restriction enzyme passes the recognition site, it's going to make a cut by breaking the bonds on the DNA backbone. There's 800 different restriction enzymes, and there's 2 different types, sticky end and blunt end, and that's just based on the way that they actually cut the DNA. There's nothing in the syllabus that asks you to talk about these 2, but I'm going to talk about it anyway. Now, just so we know with the naming, the first 3 letters of the restriction enzyme is indicating the bacterial species that it was isolated from. The 4th letter is the strain from that bacteria. And then the Roman numeral is if there's more than one restriction enzyme from that bacterial strain. So the EcoR1 is the E. coli, the R is the strain of the E. coli, and then the 1 or the I is the first restriction enzyme isolated from that strain. Okay, so a sticky end restriction enzyme looks like what you see in the image on the right. So it's going to cut the backbone at a different location on each strand in the recognition site. So it leaves a little bit of overhanging ends on the end of the fragment. So that's why we call it a sticky end, because it's got some random exposed bases that will kind of help it to bind to complementary sequences. So then those bases can make base pairs with other molecules that have the same or the complementary sticky ends. And EcoRI is an example of a sticky end restriction enzyme. They're most commonly used because they're easier to deal with. That particular enzyme cuts at the recognition site GAATTC, which is what you can see in the image on the right, and it cuts specifically between the G and the A. So in the top image, you can see the blue G and then AATTC, and it's cutting between the G and the A. And on the opposite strand, it's going obviously anti-parallel, and that sequence is going in reverse. So it's going to cut it differently on each strand. Okay, a blunt end restriction enzyme is going to cut DNA directly across the molecule cleanly, so it doesn't leave any overhanging strands. And then we've got that restriction enzyme He1III, or however it's said, and it cuts between the recognition site GGCC, right between the G and the C. So you can see in the image, it's a bit more straightforward. It just cuts right between a GGCC and leaves a blunt end. Now, these restriction enzymes are useful if you want to identify mutations and polymorphisms. I mentioned this previously in the last lesson, and this isn't specifically in the syllabus, but it's interesting. We talked about a polymorphism being an allele that has at least a 1% frequency in a population. So that's a much higher percentage than just a random mutation. So it might be a particular gene that 1 in 100 people show, which is quite common, and we call that a single nucleotide polymorphism, where it only involves one base. So you can see on the right there you've got 3 people, and the single nucleotide polymorphism is shown highlighted in that grey section there. So it's one base that's different, and it occurs in at least 1% of the population. If one person in 100 has that polymorphism, then if that polymorphism exists where the restriction enzyme would normally cut, so if it's within a recognition site, then that means that the restriction enzyme won't cut that segment in the correct place because the sequence is different to 99% of people. So what would happen is you might get a fragment that's larger in 1% of the people because the restriction site is not there, so the enzyme hasn't cut it in the right place. So researchers can use restriction enzymes to find those specific single nucleotide polymorphisms that are linked to diseases and things, because a lot of those have been shown to increase the risk of disease. Okay, so we've made a cut in our gene using restriction enzymes, and now we have our good old friend the ligase enzyme, which we've heard about before, which joins strands back together in the process called ligation, or a ligation reaction. This just helps the DNA strands to join back together. We heard about ligase with repairing strand breaks and rejoining the DNA double helix. So it basically, once a cut has been made, bases are going to want a complementary base pair back together, but the ligase assists with that process. So it rejoins hydrogen bonds and allows the strand to come back together. The conditions of the ligation reaction can be adjusted. So it depends on the type of restriction enzyme that was used. They use different temperatures and things to ensure optimum conditions for binding or rejoining of bonds. Okay, so if we have a sticky end fragment, the ligation is a lot easier because the complementary base pairs are exposed and they're going to naturally want to bond together. The ligase allows that bond to join easily, and it will allow you to add a new gene into a bacterial plasmid and seal it all back up together. We'll have a look at that in a minute. With our blunt end fragments, it's a bit tricky because the fragments can kind of join up randomly, and if they come in contact, the ligase joins them together, and often there's errors with that. Sometimes you have to use a blunt end restriction enzyme to isolate the gene that you want, but they have to do extra little things to make it work, like adding some DNA fragments to make sort of a fake sticky end so that it all joins nicely. All right, so when we're talking about recombinant DNA, I want you to look at the picture on the right to see where we're going with this. Now, a plasmid is a piece of simple circular DNA that comes from bacterial cells. We use these, and I'll talk about that in a minute, because they're simple, small, quick. so much easier to manipulate in the lab. And the purpose of recombinant DNA is to insert a target gene, so like this blue insulin gene here, into this bacterial plasmid so that we can then put that plasmid back into its bacteria so that the bacterial cell makes our insulin. So we're kind of using the plasmid as a vector to express our gene that we're wanting to express. Then that could be put into a plant cell to make a genetically modified plant. It could be put into an animal to make a genetically modified animal. So that's the purpose of recombinant DNA, and we're recombining the DNA that was in that bacteria with some new DNA from a human in this case. Okay, so sometimes we use this to clone particular genes, make more of that gene. So this is how they would have done it to make insulin for hospitals to give to people. There's other things that they use for medicine, like epidermal growth factor, which is used for burns in hospitals, interleukin-2, which is used for cancer treatments, antibodies for vaccines. So this is a technology that's used already in medicine to have a stockpile of different proteins and things that are needed for making people better in hospitals, using the actual genes in the human body that would create those proteins. So this is a plasmid. So over on the right, that's a bacterial cell, the big purple looking thing. And we know that bacterium have their chromosomal DNA, which is in the middle. So it's kind of like what we have, but it's just a big bunch of like chromatin type thing folded together. We don't use that because that's still too complex and large to insert genes into, but we are going to use these red plasmids, okay, because they're just simple sections of bacterial DNA, and we can manipulate those and put them back into the bacteria. Over there on the left, you can see that we are using those because they replicate independently of the chromosome. So the bacteria can still survive and do its thing because its chromosomal DNA is transcribing and translating, but the plasmid DNA does that separately. So it's very useful because it can be doing its thing while still surviving and making what it needs. So we call them a vector, and we're inserting target DNA in there to produce a recombinant plasmid. That is then placed into the bacterial cell. The genes can replicate, so they'll do what they would normally do in a cell, and then we're using the bacteria to produce the protein product that we're wanting. So plasmids are used as vectors because they're small, so they're easy to manipulate in a lab. They have a large range of restriction enzyme sites that are going to be useful, so the appropriate plasmid is chosen to suit the needs of the scientist. And those recombinant plasmids can self-replicate independently and quickly once they're inside the host cell. Now, sometimes they will use genes and insert them into plasmids so that they can identify that the plasmid has taken up the gene that they wanted it to. So you can see on the right there, the target gene is in blue, but they've also put in this antibiotic resistance gene. We'll talk about that later, but this is so that at the end, when they want to check if the bacteria has actually taken up this plasmid, some of them will die when exposed to antibiotics because that means they haven't taken up this antibiotic resistance gene, so they die, which means they weren't going to be useful because they didn't take on the gene of interest. The ones that did take on the target gene and have the antibiotic resistance gene will live, and so therefore you know that that bacterium has taken up this plasmid and is expressing the gene that we want it to. Another one that they might use is GFP up there at the top, and that would make it fluoresce under UV light so that they can identify which colonies are containing the target gene. Okay, so creating recombinant DNA is done in these 4 steps, which should now be making sense now that you know about ligases and restriction enzymes and plasmids. So the target DNA is cut using a sticky end restriction enzyme and isolated. So that's shown up the top here where you've got in purple, the target DNA which has been cut. We can see that it's a sticky end enzyme. Step 2 here, the bacterial plasmid is cut using the same restriction enzyme so that it has complementary sticky ends. That means that they are going to want to base pair together. So in the green, we cut the bacterial plasmid with the same restriction enzyme. So you can see that the ends of this green plasmid DNA match up with the ends of the purple target DNA. So then the target DNA and plasmids are placed together, and ligase is used to assist in rejoining the bonds. Now sometimes the plasmid closes up again, so the sticky ends of the plasmid will just rejoin and not actually like incorporate the target DNA into it. So that means it's failed, and that would be called non-recombinant. Sometimes the target DNA can go in back to front or upside down, which can affect whether the gene's going to function properly. So there are mistakes that can happen, and that's why we then go on and check that the plasmid has actually taken up the gene that we want it to. So once it's worked, here in number 3, you can see that the purple piece of our gene is inserted into the plasmid, and then it's permanently part of that plasmid now. So we call that a recombinant plasmid. So once we have our plasmid, we need to do some things to it to add it into the bacteria. So you can see on the image on the right, our DNA that we have newly created, our new recombinant plasmids, get added into the bacteria, and they heat shock the bacteria so that it takes up the plasmids. So they would do this in specific lab conditions, and it's supposed to make, as you can see on the right, the bacterium take up the plasmid into its cell. So that's called a transformed bacterium. Sometimes they won't transform at all, so they might not actually take up a plasmid at all. Sometimes they might take up a plasmid, but it hasn't isolated the gene, so it might transform but with a non-recombinant plasmid. So it doesn't always work, and that's what we talk about when we say a transformation efficiency. So you might be given some data on how many actually took up the bacterium, how many bacterium took up the gene of interest, how many took up the plasmid but without the gene, and how many didn't even take up the plasmid, and then asked to talk about the effectiveness. So if we have a bacterium here that's taken up the plasmid, we then grow that bacteria onto an agar plate, and we would have some way of identifying it, like antibiotic resistance. The ones that have lived in that case are going to contain the plasmid, and then they can go off and grow that bacteria, get it to make lots of proteins like insulin, for example. There is a video summary there, which is quite a good little summary of bacterial transformation. But this is really the process where we're making the bacteria actually take up this plasmid and produce the product that we were wanting it to produce. And that's how they check that they have actually got the correct bacterium. If they didn't check by using antibiotic resistance or something like that, then everything would grow on the plate, and then you might go off and do other things with that bacteria, and it might not even have the gene of interest that we wanted. So it's really important that they check that the transformation has worked. This is another image showing that, and they've used the ampicillin gene here as well, so like an antibiotic with antibiotic resistant gene. Here at the top left, you can see the gene of interest has been taken from a human cell, this little red piece of gene here. They've cut the bacterial plasmid multiple times or on multiple bacterium, and they've inserted the DNA fragment into that plasmid using ligase. So the one that has taken on the gene here on the left is the recombinant plasmid. One that reseals back together is non-recombinant. And so the recombinant plasmid, if it gets taken up into the bacteria, is transformed. The non-recombinant plasmid can still be transformed, but it hasn't transformed with the gene of interest. And on the right there, that means that the bacteria hasn't even taken up a plasmid at all. So it hasn't been transformed and it's not expressing any plasmid DNA. The overall aim of this technology is to get a transformed recombinant plasmid. Now, in eukaryotic cells, it has been used in crops to transfer genes into plants, like I described earlier. So that could be to produce higher yields, have different resistances to things. So they're transgenic plants and they're all grown in the lab and cultured. And they're even doing this to put in exotic genes into plants to kind of mimic expensive rare plants and make a lot of money. This is just another diagram showing the selection and screening of transformed bacteria. They've used a particular gene with their gene of interest that's going to come up as a different color. So you're going to get white indicating a recombinant plasmid and blue indicating non-recombinant plasmid, so it hasn't worked properly. So on the left, the bacteria that's been transformed with the plasmid is going to come up white, and the one that has been transformed but not with the correct gene of interest will come up white. The one that hasn't transformed at all won't grow at all on the agar plate. So it's just a way that they check that process. Okay, so now we're going to go into these technologies that are used after recombinant DNA technologies. So the first is PCR, and PCR has several uses. And in this example, we're going to talk about DNA amplification. So it's about making multiple copies of DNA. So it can be used directly after what we've just talked about with our plasmid, recombinant plasmids. But it can also be used in DNA sequencing, profiling, cloning, mutation detection. So it's quite a widespread technology, and that is one of the syllabus dot points, is the purpose of PCR. So DNA amplification is necessary when we've only got a small amount of DNA. So for example, we've made some plasmid, recombinant plasmid DNA, and we want to make more of it. And it uses polymerase chain reaction to create a large quantity of DNA that's identical to what we started with. On the right there, you can see that they would also use PCR for fossil DNA, where they only have very minuscule amounts, or they can use it in crime scenes if they've got really limited amount of DNA. Okay, so polymerase chain reaction. Polymerase is the enzyme that we've talked about in previous processes, where it's the enzyme that goes along and adds nucleotides to DNA. It works in that way. So we basically, on the bottom right there, you can see a scientist loading some DNA samples into the machine to do PCR. So they use those tiny little plastic tubes to make a mixture. The mixture contains the target DNA to be amplified. So that might be the sample from our bacterial plasmid. It might be the sample from the crime scene or the old fossilized bone. So they put the target DNA in there. They add free nucleotides, so the A's, T's, C's, and G's, to build new strands. They add DNA polymerase, and then they add 2 DNA primers, which are complementary to the ends of the target DNA. So they're kind of like little markers to specify the start and end of the DNA fragment. Don't worry too much about that, because remember, you just need to know the purpose of PCR. They put it into a machine, and then you can see in the table there on the right, it does a number of cycles depending on what the scientist puts in. And every cycle, it does an exponential growth of the original DNA. So if they don't do any PCR, 0, then they get 1 DNA strand. If they do 1 cycle, they get 2. 2 cycles, they get 4. 3, they get 8, and so on. So they could chuck it in for a few hours and get millions of copies of that original DNA. So that's what that looks like on the left there. They basically do a series of cycles in this DNA thermocycler, and it basically changes the temperature quite systematically in order to break bonds and reform bonds. So in the image on the left, if you have a look at the target sequence there, As we move down, step 1 is denaturation. So we're heating briefly to separate the DNA strands. So you can see the two separate strands. We're then cooling to allow the primers to bond to the ends of the sequence. So that's called annealing. And then the DNA polymerase then extends along and adds nucleotides. So very much like we saw with our DNA replication, but mimicking it in a lab environment. One cycle gives us 2 new DNA strands. The second cycle does the same thing. So it heats again, breaks the bonds, reforms. Next cycle, heats, cools. Okay, so you can see the denaturing happens at 95 degrees and breaks the hydrogen bonds. The annealing, the temperature is reduced to 50 to 60, and then extending, it's increased to 72, which is optimal for the polymerase to bind and move along and add its bases. So that's repeated up to 50 times to ensure there's sufficient target DNA, and scientists will do that to make sure they have a whole stack of that DNA so that they don't have to go and do the recombinant DNA again. Okay, now electrophoresis is another technique which is used to visualize the fragments of DNA that we are getting from different samples. So it could be used in crime scene analysis where we've got multiple suspects. They will digest the DNA using restriction enzymes and break the DNA up into specific fragments, but that will be different depending on different people because of whether or not they have single nucleotide polymorphisms. So in that case, different people will have fragments of different sizes, and they run the DNA through this gel, which separates the strands of DNA based on the size of their fragments in base pairs. So I'm going to go through the steps of that process. The electrophoresis gel is prepared, which is made of a jelly-like substance called agarose, and it makes a porous matrix. So it's like a microscopic little network that stuff can move through like a big microscopic sponge. The gel is placed into an electrophoresis chamber and it has a charge put through it. So there's going to be the wells on one side, which is where you're going to load your DNA samples, and they're going to migrate towards the positive end of the electrophoresis chamber. Because of the charge of DNA, they're going to naturally want to move to that positive end. Each DNA sample, so maybe the 3 different suspects or the 3 possible fathers of the child, are going to be treated with restriction enzymes to chop up the DNA into fragments. And then you're going to load that DNA into the gel by using a micropipette into these wells 1, 2, 3, and 4. Over here on the far left, they put a sample called a DNA ladder, which is a mixture of DNA that has fragments of known sizes. And so that will separate at very specific sizes. So you can see here 1,500 base pairs, 1,000, 900, 800. And that gives us an indication to compare to our sample DNA. The gel is covered with a controlled pH solution, so a buffer, which is just an ionic substance that allows electrical current to flow through the gel. The power is turned on and the DNA migrates towards the positive end. And as you might have learnt in chemistry, the smaller fragments are going to travel further because they're small and they can make their way through that molecular network. And so the little bars are going to sort themselves out according to their length. So you can see here, person number 1, there's a fragment at just under 1,500 base pairs, one between 8 and 900, another one at 5, one at 350, one at 2, and one at maybe 175. So you would be able to estimate the actual fragment lengths. Now you can see person 1 and 2 have really similar fragments there compared to 3 and 4. So that could give you some indication as to, I don't know, who's got similar. Maybe person 1 is the child and they're comparing it to 3 possible daddies, and person 1, the child matches the first dad mostly. So that's how that would work in that particular example. The DNA fragments can then be detected afterwards by taking the gel out and viewing it under a particular, often they use like a UV light to visualize the fragments, and they can take special photos to visualize the electrophoresis results. Okay, that's just another depiction of that. You can see on the left there, the DNA sample gets loaded into those wells. You can see that the large fragments are closest to the wells and the small fragments are further away, and they're migrating based on electrical current. And that's what it looks like compared to the DNA ladder on the very far right, which tells you what the base pair sizes are in comparison to our samples. Okay, so it's commonly performed after DNA amplification to allow scientists to separate the different DNA fragments or proteins present in a sample based on size. That's used for DNA profiling or isolating fragments for further use. So we might want to isolate a particular fragment. We might know that the gene that we're trying to look for its role in a disease is 1,000 base pairs. So we know that that particular bar would correspond and be the gene that we're looking for. All right, so that's the overview of electrophoresis. Again, you need to know the purposes of these techniques, not the ins and outs of how they work. So gel electrophoresis is used for comparing DNA. It could be used for DNA screening, so comparing against a control. It could be for paternity testing, crime scene analysis, comparing between species. It can be for confirming that you've actually isolated the correct gene in PCR. So like I said, if you were looking for a gene that's 850 base pairs, you can run your electrophoresis and hope that your bar comes out at 850 against the DNA ladder. You can also identify DNA fragments obtained using restriction enzymes. Now over there on the left, that's what I was talking about, where you view it under a particular stain. So it shows up as pink fluorescent bands, and then they can actually chop out the bars that they're wanting to obtain. Okay, so with appraising, you would be given things like transformation efficiencies, like we just talked about with our bacterial transformation. When researchers are promising impressive outcomes with biotechnology, the idea of appraising is knowing how likely they are to succeed and weighing that up against the risks. So that would be the types of things that you might be asked, and that's also mentioned in the syllabus guidance notes. Now, there's a really good resource on Khan Academy, the link's there, and it gives you a really good summary of all things to do with recombinant DNA, and there's some good practice questions on there. So you need to be able to describe the process of making recombinant DNA, isolation of DNA and cutting using restriction enzyme, insertion of DNA fragments using a plasmid vector, joining of DNA using ligase, and amplification of that DNA via bacterial transformation. That is probably the process that you need to know the most, because it wants you to describe that process. You need to explain the purpose of PCR and electrophoresis and appraise data to determine success rates. Remember, the current biotechnology techniques should include electrophoresis, PCR, or CRISPR, which was last lesson. And there's some examples there of data that they might ask you about for appraisal.